The clinical symptoms of Campylobacteriosis occur usually between 2 to 5 days after exposure. There may be lower abdominal pain, watery diarrhea, which may be bloody, nausea, vomiting, and fever. The symptoms are usually self-limiting and resolve spontaneously anywhere from a day to a week. Some infected individuals are asymptomatic. In rare cases, a chronic neurodegenerative disease can develop 2 to 3 weeks following the initial infection. Reactive arthritis with painful joint swelling may also occur, which is strongly associated to individuals with the MHC1 tissue type of HLA-B27. The primary reservoir of Campylobacteriosis is animals. This disease is a zoonosis, transmitted mainly through direct contact with infected animals such as poultry, for example chickens, or other livestock such as cattle, and pets, for example dogs. It can also be transmitted indirectly through consumption or processing of contaminated chicken food products, drinking inadequately chlorinated water, or unpasteurized milk. Campylobacter has a low infectious dose. Just a few hundred bacteria can cause an infection. This graph shows the relatively stable national Canadian rates between 25 and 29 cases per 100,000 population, which corresponds to between 8,500 and 10,500 cases per year. The rates in Ontario, indicated by the purple line, are also fairly stable, hovering between 23 and 30 cases per 100,000, which is on average around 3,500 cases a year. The rates in Toronto were slightly higher than both the provincial and national rates, at between 24 to 38 cases per 100,000, corresponding to between 750 and 1,000 cases a year, but in the last 2 years the numbers have dropped to provincial levels. It's difficult to perceive that a gastrointestinal disease may be associated with an occupational health risk. However, one of the largest reported Canadian outbreaks of Campylobacteriosis was associated with occupational or recreational exposure. Nearly half of the participants in a mountain bike race in rural British Columbia had diarrheal illness, and 25 of them were confirmed to have Campylobacteriosis. The source was likely from animal manure that contaminated the mud which covered many of the bike racers, who did not have time to wash their hands while they ate and drank along the race. A case of Campylobacteriosis with or without symptoms is confirmed in the laboratory by the isolation of Campylobacter from an appropriate clinical specimen, and a probable case is a person with clinical illness who is epidemiologically linked to a confirmed case. Fecal specimens need to be shipped in transport media if the transit time is more than 2 hours, as the bacteria are relatively fragile and sensitive to environmental stresses, such as drying, heating, disinfectants, and acidic conditions. Campylobacter culture requires a microaerophilic atmosphere with 5% oxygen, 10% carbon dioxide, and 85% nitrogen. An evacuated anaerobic jar with gas generator packs is usually used to produce this mixture of gases. Special selective media with charcoal or blood is used, and antimicrobial agents added to inhibit the growth of normal gut flora with an incubation temperature of 42 degrees Celsius. Campylobacter jejuni is a gram-negative curved rod with a corkscrew shape with bipolar flagellae. It is a microaerophilic bacteria and requires reduced oxygen concentrations that are lower than atmospheric concentrations to grow. You can see pictures of stained Campylobacteria in the picture on the top, and a scanning electron microscopy picture on the bottom, clearly showing the flagellae at both ends. Campylobacter colonizes the intestinal mucosa by adhering to endothelial cells, then gaining entry through endocytotic vacuoles. It is hypothesized to have an enterotoxin, but its virulence mechanisms are not well understood. Other human pathogenic Campylobacter species include Campylobacter coli. Antibiotics are not usually used in treatment, as the disease is self-limiting and therefore only used when illness is severe or prolonged. There is some resistance to beta-lactam antibiotics and antibiotics in the macrolide group, if used early in the infection, may reduce the length of illness. There is no vaccine available for human use. Adequate public health control measures include the investigation and follow-up of outbreaks, for example, association with consumption of unpasteurized milk from the same supplier, as well as prevention of further exposure to vehicles such as contaminated food or water and reservoirs of infection such as infected livestock. Let's now focus on a large group of small, non-spore-forming gram-negative rods called Enterobacteriaceae. These bacteria are ubiquitous in water, soil, and vegetation. They're also present as normal flora in the large intestines of animals, including humans. Enterobacteriaceae includes several genera of bacteria. Some are opportunistic bacteria, such as Escherichia, Klebsiella, Serratia, Enterobacter, Citrobacter, Proteus, Morganella, Providentia, and others are pathogenic bacteria, such as Salmonella, Shigella, and Yersinia. They all have three types of antigens. The O antigen is the repeating polysaccharide units of lipopolysaccharide on outer membranes of the cell wall. The H antigen is the flagellae protein, and the K antigen is the polysaccharide capsule. There are many different serotypes with different O, H, and K types, for example, E. coli 0157H7. Enterobacteriaceae can also be differentiated into lactose fermenter and lactose non-fermenters. This can be determined using McConkey agar, which is both a selective and differential medium. How many of you remember this medium from ENH 222? As a reminder, McConkey agar contains bile salts and crystal violet, which inhibits the growth of gram-positive bacteria. McConkey agar also contains lactose and a dye that turns red at acid pHs generated by lactose fermentation. This differentiates lactose fermenters from non-fermenters. A pink to red color is produced by lactose fermenters, such as Escherichia and Enterobacter, and lactose non-fermenters are colorless or transparent colonies, such as Salmonella and Shigella. Yersinia is also a lactose non-fermenter. Salmonellosis is sometimes confused as food poisoning because it has a very short incubation period ranging between half a day to 3 days after infection. There is usually non-bloody diarrhea, abdominal cramps, nausea and vomiting, fever, muscle aches, and headaches. Illness may last up to a week. It is common in North America and is transmitted through contaminated food such as raw eggs and milk. Pets, especially reptiles such as turtles and birds, can produce large amounts of salmonella. Human-to-human spread is possible, but less likely as it has a high infectious dose of more than or equals to 1,000 bacteria, and animals tend to shed more bacteria in their fecal matter. In the past 10 years, all the national, provincial, and municipal rates for salmonellosis were very similar, between 15 and 26 cases per 100,000 population. In terms of numbers, this corresponded to around 7,000 cases in Canada, 3,000 cases in Ontario, and 600 cases in Toronto each year. There have been some interesting salmonellosis outbreaks. The latest ones occurred this summer, and one was a massive outbreak with more than 500 confirmed cases of Salmonella Newport. The outbreak was associated with exposure to red onions imported from the US. The second outbreak also occurred during the same time period. It was a smaller outbreak, and most of the cases were in Ontario. The implicated item was imported peaches from the United States. In 2016, there were 2 outbreaks in Toronto, both in September. We don't have any further outbreak information later than this because Toronto Public Health stopped publishing their annual reports. The first was linked to the consumption of chicken shawarma at a food premise located in Toronto, and the second associated with pork ribs and other cross-contaminated items at a private party in a Toronto home. A case of salmonellosis with or without symptoms is confirmed in the laboratory by the isolation of Salmonella species from an appropriate clinical specimen, and a probable case is a person with clinical illness who is epidemiologically linked to a confirmed case. Salmonella is isolated from fecal specimens by culture in enriched broths using selective media that inhibits Salmonella typhi, the species that causes typhoid fever, which we're not detecting since we're specifically detecting other Salmonella species that cause salmonellosis only. The selective and differential media, McConkey agar, is used to identify a lactose non-fermenter. The API multi-mini-test biochemistry panel is also used to further confirm the identity of Salmonella, which is a lactose non-fermenter, since there are other lactose non-fermenters that cause gastrointestinal disease. Serotyping is also carried out to determine the O and H type using slide agglutination assays with specific antisera. There are more than 2,000 serovars or serotypes of Salmonella enterica. Examples are Salmonella enterica serovar typhimurium, also known as Salmonella typhimurium, and Salmonella enterica serovar enteritidis, also known as Salmonella enteritidis. There are different serotypes have a very wide host range. Many strains are host adapted, and Salmonella typhi, which causes typhoid fever, and not salmonellosis, is only found in humans. The pathogenesis and virulence of Salmonella is associated with a number of secreted proteins, such as the invasion and surface protein antigens, which are coded for by the IMV and SPAR genes. These proteins are delivered through the type 3 secretion system. You've heard of this secretion system before in Lecture 3, when the plague bacteria Yersinia used this delivery system to kill macrophages. This causes stimulation of membrane ruffles, allowing the bacteria to gain entry into host epithelial cells and invade the submucosa to reach the lamina propria, causing intense inflammatory responses from the host. Salmonella can multiply in macrophages. Treatment is usually supportive with rehydration to replace fluid and electrolyte loss from severe diarrhea. Antibiotics are not usually prescribed, as they may prolong illness and increase carrier status. They're only used for cases with underlying medical conditions to prevent systemic spread. There is no vaccine for salmonellosis in humans, so prevention is through improved hand hygiene, especially after handling infected animals, and thorough cooking of poultry, meat, and eggs. Adequate public health control measures such as investigation and follow-up of foodborne outbreaks and recalls of contaminated food items helps to prevent further outbreaks, as well as prevention of further exposure to reservoirs of infection. The next reportable gastrointestinal disease we'll look at is shigellosis. This disease also has a short incubation period between 1 to 2 days. Shigellosis produces dysentery, which is inflammatory colitis with cramps and frequent small volume stools with blood and pus. Unlike salmonellosis, shigellosis does not spread outside the intestinal tract. In rare cases, there may be a condition called hemolytic uremic syndrome, where the kidneys are affected, which is a condition that is also sometimes present in E. coli 0157H7 infections, and reactive arthritis with painful joint swelling, which is also sometimes present in Campylobacteriosis. Unlike salmonellosis, shigellosis is only transmitted from one human to another, as it is only found in humans. This is via the fecal-oral route. It has a low infectious dose, and less than 200 microorganisms will cause an infection. A secondary attack rate is quite high, at just below 50%. Indirect transmission can occur through exposure to contaminated food or water. You can see from this graph that the incidence rate of shigellosis in Canada is fairly low, at between 2 to 3 cases per 100,000 population, corresponding to over 600 to 1,000 cases each year. Ontario rates are also within the same range, and there are an average of around 250 cases of shigellosis a year in this province. The rate in Toronto is twice as high as the national and provincial average, between 3.5 and 6 cases per 100,000, corresponding to 100 to 150 cases each year. A case of shigellosis with or without symptoms is confirmed in the laboratory by the isolation of Shigella species from an appropriate clinical specimen, and a probable case is a person with clinical illness who is epidemiologically linked to a confirmed case. Unlike other gastrointestinal bacteria, Shigella have no flagellae, so they have no H antigens and are not motile. They can be cultured using selective and differential media such as McConkey agar and are lactose non-fermenters, so they grow up as colorless colonies. There are a number of serogroups, some of which are more prevalent in North America than others. The most common one is Shigella sonii, which can be identified with slide agglutination assays. The API multi-mini test panel is also used to further confirm the identity of Shigella, as it is a lactose non-fermenter and needs to be differentiated from other lactose non-fermenters such as Salmonella. If we compare the results with those from Salmonella, we can see that there is quite a bit of difference, especially in the first half of the strip, where only one test yields a positive result, whereas Salmonella produces positive results for 5 of the 10 tests. Like Salmonella, Shigella also invades the intestinal M cells. It also has invasion protein antigens, which are also delivered through the type 3 secretion system, just like the Yersinia yaps and Salmonella invasion and surface protein antigens. This invasion protein antigen causes skeletal reorganization in the host cell and enables the bacteria to enter the cell. It then polymerizes the host cell actin, using this protein to form a tail of flagellae-like structure which gives it motility, allowing it to invade adjacent cells after the bacteria multiplies. The invasion progresses till the bacteria are able to reach the lamina propria, causing an acute inflammatory response. Shigella produces a toxin called the Shiga toxin. Some species may cause fatal infections. There may be hemorrhagic colitis with inflammation of the colonic mucosa and hemolytic uremic syndrome, in which circulating Shiga toxin binds to renal tissue, causing glomerular swelling. Deposition of fibrin and platelets clog the kidney filtration system with blood clots and result in kidney failure. Kidney dialysis is required to save these cases. Antibiotics is required for treatment of serious cases, and some drug resistance is present, so second-line antibiotics may need to be used. Since there's no approved vaccines for shigellosis, provision of adequate sanitation and hygiene to control contamination in endemic countries is required to prevent transmission. Although most E. coli in our intestinal tract are harmless, there are 4 different types of pathogenic E. coli that can cause disease even in healthy individuals. We'll only have time to cover one of them, and this is enterohemorrhagic E. coli, which has caused several foodborne and waterborne outbreaks in North America. Enterohemorrhagic E. coli is also known as verotoxigenic E. coli or Shiga toxin-producing E. coli, depending on the laboratory tests that are used to detect it. The most commonly reported Shiga toxin-producing E. coli in North America is E. coli 0157H7. E. coli 0157H7 causes bloody diarrhea, hemorrhagic colitis, and hemorrhagic uremic syndrome, resulting in kidney damage and renal failure, like some types of Shigella. It also has a low infectious dose, approximately 100 bacteria can cause an infection. E. coli 0157H7 is an animal reservoir, mainly cattle, so ground beef from infected animals, if undercooked in contaminated hamburgers, can result in infections. Exposure to contaminated water, unpasteurized milk, and fruit juices have also been associated with outbreaks of this pathogenic E. coli. We've already learned about the Walkerton outbreak earlier on in this semester. In the past decade in Canada, the incidence rate of verotoxigenic E. coli infections has been quite stable and low, between 1.5 to 3 cases per 100,000 population. This is around 500 to 1,000 cases per year. In Ontario and Toronto, the rates are lower than the national rates, at slightly lower than 1 to 2 cases per 100,000, which is around 150 cases a year in Ontario and about 30 cases a year in Toronto. Recent outbreaks of verotoxigenic E. coli include one in 2016 with only 9 cases, 3 of which were in Toronto and the rest in Ontario. There were 2 outbreaks in 2015. One was a national one with only 3 cases in Toronto. Once again, no common sources were identified. A second outbreak in Ontario was associated with pig roast, and 2 of the 6 cases were located in Toronto. A case of verotoxigenic E. coli infection, with or without symptoms, is confirmed in the laboratory by the isolation of verotoxin-producing Escherichia coli from an appropriate clinical specimen, and a probable case is a person with clinical illness who is epidemiologically linked to a confirmed case with hemolytic uremic syndrome. Enterohemorrhagic or verotoxigenic E. coli have an outer membrane adhesion that enables them to attach to host cells. This causes attaching and effacing lesions which destroy the intestinal brush border, resulting in diarrhea. They also produce shigellotoxins that are similar to the ones produced by Shigella, which can cause hemolytic uremic syndrome. In young children, antibiotic treatment may increase the risk of developing hemolytic uremic syndrome, which then requires hemodialysis and treatment in hospital. Fecal specimens are submitted to the lab for testing, and E. coli 0157 can be concentrated using immunomagnetic beads that are coated with 0157 specific antibodies. They're also cultured on a special type of MacConkey agar with sorbitol instead of lactose. Enterohemorrhagic E. coli cannot ferment sorbitol, so they grow as colorless colonies, whereas other types of E. coli grow as pink colonies. The bacterial colonies need to be confirmed using 0157 antisera. To detect verotoxigenic E. coli, which is another laboratory test for enterohemorrhagic E. coli, culture supinatants are added to vero cells and observed for cytopathic effects. If you look at the figure via the web link, you can see that the vero cells are completely destroyed after 24 hours. The API multi-mini test panel is also used to confirm that the bacteria are E. coli. You can see that the results are quite different to Salmonella and Shigella, especially in the first half of the strip, where 4 of the 10 tests are positive. I hope you remember getting a positive indole test in your ENH 222 lab experiment, as this result differentiates E. coli from Salmonella and Shigella.